Hello everyone. Today we will see about some important discoveries related to the DNA and salient features of the DNA double helix. Firstly, we will see the important discoveries. Frederick Fisher identified the DNA as acidic substances present in the nucleus and he named it nuclein. James Watson and Francis Crick in 1953 proposed double helical model of DNA. James Watson and Francis Crick proposed the double helical model of DNA. This structure of the DNA based on the X-ray diffraction data given by the Maurice Wilkin and Rosalind Franklin. Irving Kirchhoff in 1944, before the James Watson and Francis Crick, observed that the ratio between adenine and thymine and cytosine and guanine ratio between adenine and thymine and between guanine and cytosine are constant and equals 1. Ratio of adenine and thymine will be equal and constant, and ratio of between the cytosine and guanine will be constant and equals. Now come to the salient features about the DNA double helix. In the double helix, two strands of DNA present. These two strands are made up of polynucleotide chain. These two strands have anti-parallel polarity. If one strand runs from upward to downward, other strands move from downward to upward direction. The direction of the movement of the strands will be from 5' to 3' direction. Other strands will be from downward to 5 to 3' direction. Not in the same way, opposite will be one strand if will be in this direction, other strands will be in this direction, but will be parallel. Backbone of the strands made up of sugar and phosphate. Backbone of the DNA double helix made up of sugar and phosphate. Inside of the backbone, nitrogen bases projected. Inside of the double helix present nitrogen bases. On the boundaries or on the strands, what present sugar and phosphate in both the strands. So sugar and phosphate form the backbone and nitrogen bases projected inward, inside. Bases of the two strands paired through hydrogen bonds. Bases of the two strands paired through the hydrogen bond. Between the thymine and adenine will be double hydrogen bond, and between cytosine and guanine present triple hydrogen bond. In the double helical structure of the DNA, the pitch or one turn cover the distance of about 3.4 nanometer, and distance between the two base pair is of 0.34 nanometer. 10 base pairs, about 10 base pairs present in one pitch or in one turn. The coiling of the chains, both the chains are in right-handed fashion in this way, not in the left-handed fashion. In the right way in this direction, not in the left-handed fashion. The plane of one base pair stack over the other, and this stacking of the base pair with the hydrogen bond, stacking of the one base pair over the other base pair provide the stability to the double helical structure of DNA. Stacking of base pair over the other base pair with the hydrogen bond provide the stability to the double helical structure of DNA. So this is about the salient features of DNA.